I begin our third part, which will talk about focus on cinema and theater and narrative to a certain extent. And my talk today will focus on special neuronal elements that allow us to perceive theater and cinema, specifically the perception of the other in cinema and theater. And I will elaborate on that during my talk. And in the second part, Alon will continue with more high order, more cognitive level functions that allow us to perceive these forms of art. So I would like to start with fish, not with humans. So basically, is this ability to observe others and enjoy interaction with them is specific to humans or not? So in this study in newborn guppies, these are not guppies, but I didn't find a good picture for that. So in the case of guppies, if you put 2 weeks old guppies in an aquarium, they prefer to spend more time during more time in the part of the aquarium that is adjacent to a transparent glass with other swimming fish around them on the other side of the glass, or near a mirror. So they spend more time if there is something moving, possibly conspecifics, near them. And as they mature, the specific patterns of what they prefer, which moving fish they prefer, changes. So in female adult guppies, if you put a female in an aquarium and there is a partition in transparent glass, and on the other side there are males, the female will spend about 80% of the time near this transparent glass. So the female sees the male and the male sees the female. But if you put a one-way glass so that only the female will be able to observe the male, but the male doesn't see her, so the interest of the female in that sort of observation drops. So only in 60% of the time, the female will spend near this transparent glass. And if you replace a live male with a video playback of a male in the aquarium, it wouldn't matter. So the female prefers to the same extent live animal and a recorded animal, as long as they don't interact. But of course, it prefers more live interactions. So that says something about us in a way. So often we prefer to go to theater or to the cinema instead of going and having a real bidirectional interactions. But that's not the case in fish. So things I will talk about during the lecture will concern with biological motions and the observations of conspecifics, facial recognition, which is something very important in humans and for theater and cinema in particular, and the neural basis of imitation and empathy. So just when we talk about biological motion, let's start with the definition of what is theater and cinematography. So theater from Greek is theatron, a place for viewing, itself from the verb theaomai, which means to see, to watch, to observe. Cinematography comes from kinema, movements, and graphein, to record. So I'll argue today is that that it's true that theater and cinema relies on the observation of movements of conspecific, but there are much more than that, and that's something that we'll Alon elaborate on it too. So motion. There are many brain regions that are dealing with motion, and in the visual cortex there are many parts of it in which cells respond to moving stimuli. In particular, V5 was suggested, an area of the visual cortex was suggested to be involved in motion and perception of depth, something that we talked about a few weeks ago. So when a monkey is is shown a screen with moving dots in a specific coherent pattern, this moving pattern evokes a perception of a revolving cylinder. And the area V5 is considered to be important for that sort of sensation. But this is not biological motion. This is just movements. What about biological motion? So the pioneer in the field of biological motion was the Swedish scientist Johansson, and he made these Johansson moving dots, moving biological dots. So the way he did it, he put LEDs on human subjects with black shirts, with black costumes, and recorded them in complete darkness. That's how it looks like. Just with having 14 moving dots, we can perceive the movement. For example, here it's a jumping figure. And subsequent experiments show that you can perceive the gender or the age of the person. This was probably an older person. And even the specific identity of the person. So, and this is all just with 14 dots. So the human brain is very, very good in identifying biological motions. And in fact, infants already at 4 months age old prefer biological motion to random patterns. So they prefer to stare at the screen which is projecting biological moving dots versus random dots. And other animals such as cats also are able to perceive this sort of moving dots in a biological, meaningful way. So which brain area supports biological motion? Something that, of course, motion pictures heavily rely on. Otherwise, we won't be able to perceive them. And indeed, there are certain subjects that are unable to perceive motion with damage to this area. So an area that is involved specifically with biological motion is called the superior temporal sulcus, located here. And in an area adjacent to it, in monkeys, there were single neuron recordings that identified neurons that respond specifically to moving figures and to patterns of biological motions. So this is a response of a single neuron responding either to human figures moving in different directions or to biological motion stimuli. So this neuron preferred a human figure moving leftwards. You see the activity. But even strongly, it fired to a pattern of biological motion corresponding to a human figure moving leftwards. You can see the strong discharge. So these neurons really perceive these moving dots patterns as something meaningful. And the neuron will fire less in opposite directions. So movie industry often challenges us with different biologically relevant movement patterns. Not necessarily for the whole body, but sometimes for individual parts of it. So in this scene from Polanski's movie Repulsion, we can almost decipher what she's writing here on the glass by looking at the expression and the movement of her eyes and the lips. It's not really lips reading because she's not saying the words, but just looking at the expressions and the movements, we can get a lot of information, and we extract information not only for facial movements but also movements of other body parts, even if they are presented in a cartoon-like fashion. So of course, in this marching hammers scene, we can also get the impression of moving legs, and also looking at specific interactions and communication through various body parts, we can understand a lot about the behavioral state and the emotional state of the actors. So, what areas in the body responds to activation of movements of specific body parts? So, if you put electrodes on the human skull and record activity of from individual electrodes, you can study which types of movements evoke a stronger or a weaker activity in individual electrodes. So it turns out that in several electrodes, you can get a preferential activation for certain movement types. For example, in this case, this posterior temporal electrode, roughly corresponding to the STS areas that I showed you before, has stronger activation. So actually the activation here it's plotted in an inverse way. So stronger is a more negative response. When the subject sees a video clip of a person moving his eyes away from the subject, in comparison with a movie clip where just a straight eye gaze is observed. And this can be also elicited just by looking at eyes specifically. So when we move the eyes away from the subject, this activity increases. And also if you open the mouth as compared to closing the mouth. Not only facial expressions evoke specific responses in this brain region. So for example, this frontocentral electrode responds to movements of legs or on hands or of hands. In this case, if the clip shows a subject moving forward, the electrode is firing, this response is stronger than for a subject moving backwards. Or for, in case of the hand, and closing of the fist is strongly affecting this electrode than opening the hand. And this might do with the fact that that sort of movement, so opening of the mouth, step forward, or closing a fist is associated with aggression. It's a threatening movement, so probably the brain responds more to this sort of threatening movement patterns in conspecifics. Other brain regions, actually a region located near, or some consider it part of the STS, the extrastriate body area, was found to respond in humans to the pictures of various body parts or whole figures. So this area responds to different body parts, photographs of them, or just cartoons, to whole figures, photographs, or line drawings, or just silhouettes, to a certain extent to faces as well. Although this is not the only areas that respond to face, and that we will talk about in a bit. But it didn't respond to any of these stimuli, tools, just scrambled figures, and animals, mammals. So this area really responds to specific features concerning with human bodies. So that's sort of an overview how we perceive general biological movements and how we recognize conspecifics. But very important feature in recognizing the other is, of course, facial recognition. We mostly recognize the other not by the body, but by its face. And of course, this is something very important in theater and cinema and paintings. In the case of theater, in Commedia dell'Arte was based on archetypical characters, each of them having a specific mask, a typical mask. So they were even called the masks. And each character was playing a certain role according to the mask that he was wearing. So Paul Ekman studied facial expression, and he identified 6 to 7 major categories of facial expression in humans. So happiness, surprise, fear, anger, disgust, and sadness. And in a series of seminal works, he showed that these facial expressions are universal across humans of different cultures. And of course, we probably inherit this ability to express emotional and behavioral states through facial expression from monkeys, which also have a wide repertoire of facial expression, and the ability of actors to move and convince us in their acting is very much based on this skill and talent of expressing the character through the face. So face recognition. As Alon said and Doris said in previous lecture, we are very good in recognizing faces. We almost tend to recognize a face even there is no real face there. But we are much better in recognizing faces when the faces are presented upwards. Subjects, and this is different from object recognition. Probably face recognition is done in a separate way than just general object recognition. And in fact, subjects who had brain damages, for example, the famous subject CK, who couldn't recognize objects, he was still able to recognize faces. And in the case of Archimboldo's fruit or other types of Archimboldo's paintings, he was able to see a face. He was saying this is a face, but he couldn't say that it is made of vegetables. So object recognition is different from face recognition. And in fact, there are few brain regions that are specialized in specific recognition of faces. The most famous one is the fusiform face area located here. That's you can see the red parts here. It's in the base of the temporal lobe. Some face responses are observed also in the STS, these areas that I talked before with regard to biological motion. But the STS areas are more involved, as you saw, with some sort of movement of the face or gaze direction, while the FFA area recognizes the face itself. So what is important? What is important in facial recognition? And these are famous study by Albert Yelbus, a Russian physiologist, who put a sort of suction cups on the eyes of subjects while they were presented with different photographs or paintings, and the movement of the eye was tracked. So you can see that mostly we tend to look at the eyes, a little bit in the eyes, the mouth, less at the nose, and a little bit in the general contour of the face. So these features are probably the most important for face recognition. And studies in monkeys, single neuron recordings in monkeys, confirmed this assumption that eyes and mouth are very important for facial recognition. So when a single, these are single neuron recordings in monkeys' face patch areas. So this neuron typically responds to a face. In this case, a drawing of a monkey face, but it also responds to a cartoon of a face. Even more than that, the response to the cartoon is stronger than the response to the actual monkey face. And it's important for this neuron to fire. It is important that the stimuli will contain both the eyes, the mouth, and the contour of a face. So if you just put eyes, the cell would not respond. If you just put a bar representing a mouse, it will not respond. If you omit the contour, so you just make a square shape without clear border, the cell will not respond. The contrast between the inner part of the face and the outer part is not important so much as well, but in order for the neuron to fire, the eyes and the mouth should be darker in contrast than the inner parts of the face. And later on, Doris Tsao, in a series of studies, identified different face patches in monkeys. Each one is dealing with slightly different features of facial representation. And so this is sort of mean response activity in one of these face areas. So if this area is presented with faces, these are different categories. So you can show this area faces, bodies, fruits, gadgets, hands, or just other stimuli. So these neurons respond specifically to faces, very, very repeatably. And the only cases when they respond to a stimuli from the fruits or the gadgets category are these two cases. In this case, they were shown an apple. In this case, a clock. So these are round objects that probably look a bit like a face. But generally, even to this face-like objects, the response was much weaker than for the face. So these are really face-specific areas. And consistent with the findings that we respond to Archimboldo's or other faces less, we recognize it with more difficulty if it's inverted. So also these neurons, when a cartoon face was presented in an inverted manner, this cell didn't fire too much. So these studies sort of emphasize the notion that we perceive the face holistically. So we don't perceive individually the eyes or the mouth. We perceive a face and the different elements in face as part of one single image. And in this famous Margaret Thatcher illusion, this notion is emphasized. So we look at the face as one whole thing, and that's the reason that we don't notice that the different elements of the face are actually distorted, because we represent it probably in a holistic manner. So I hope that Margaret Thatcher scared you a little bit. One brain region that is dealing with fear and emotional expression of faces is the amygdala. Alon will talk, actually will not talk about it later on. The amygdala is sort of emotional center, one of the emotional centers of the brain. And it was shown that different facial expression elicit different responses in the amygdala. And specifically, it's responding strongly to fearful or frightened facial expressions than to neutral faces. And if you look what spatial frequencies of the images elicit a stronger response in the amygdala, you find that high spatial frequencies are not that strong. This is shown here in eliciting a response in the amygdala, but low spatial frequencies, or in other words, blurred faces, elicit a stronger activation, specifically if the face is fearful. And this might, in a way, this amygdala response to blurred faces might explain why we often have kind of anxiety feeling when we watch blurred movements, blurred faces in movies. So this face, although it's pretty neutral, this blurry, ghosty effect elicit, probably at least in me, some slight fear response. So that might be one of the explanations to this phenomenon. So So we spoke about biological motion, observation of conspecific, and facial recognition. But as I said in the beginning, cinema, theatralic and cinematographic experience is much more than recognition, than observation of motion. Because obviously there is an empathic and emotional component involved when you watch a play or a movie. So, just to explain what I mean, I brought you this. So, I spent the whole Thursday evening looking for painful clips, and that was a suggestion by a lab member. I will not tell who. So we often empathize with another pain. After preparing this lecture a couple of times, my empathic response to this clip diminished significantly. Okay. So my idea was that you will empathize in this case, but your empathic reaction was kind of inverse. But we all know that we often have this feeling that if something painful is happening on screen or just in life, we really feel the pain almost if it's our own flesh is being cut. This study addressed this question. So how we empathize and perceive the pain of others. So in this case, the subjects were presented with photographs, with images, other painful images. So for example, a hand being crushed by a door, or a finger being cut by a knife, or just neutral, non-painful images. This will be, I will not show any more of that, I promise. The different brain regions were examined for correlation with the contrast between the painful versus the non-painful images. And it turned out that specifically the anterior cingulate cortex and the anterior insula, shown here, have a stronger response to painful images. And these two brain regions were already known as mediators of self-awareness and to a certain extent of empathy. And indeed, pain, really physical pain that subjects reported in other studies elicited a response in this area. So what was new in this study is that also a pain of others elicited a response in the very same areas. And moreover, in the case of the anterior cingulate cortex, the subjective pain index that the subjects reported, so they reported that this is less painful than this, for example, correlated with the degree of activation of the anterior cingulate cortex. So these regions perceive pain and also empathize with the pain of others. And this idea that there are kind of innate responses that allow us to perceive the pain of others, empathize with the others, and perceive our own pain and misery were in a way discussed by Nietzsche, who said that the truth is, in the feeling of pity, we are, to be sure, not consciously thinking of ourselves, but are doing so very strongly unconsciously. An accident which happens to another offends us. It would make us aware of our impotence and perhaps of our cowardice. If we did not go to assist him, well, I'm sorry, or an accident and suffering incurred by another constitutes a signpost to some danger to us, etc. So basically, this idea that that seeing a pain in another is sort of an alert sign for us makes sense that in the way that same regions, brain regions, are involved in perceiving another pain and self-pain. But another form of empathy is embarrassment. Embarrassment, a vicarious embarrassment towards another. So it often happens in movies and plays. That sometimes we empathize with the embarrassment of the actor, or sometimes just because the actor is playing badly, or sometimes also the lecturer. In this study, the subjects were presented with different scenes, embarrassing scenes. So in this case, the story was that imagine that you're in the post office and somebody in front of you is bending down to take a parcel, and his pants ripped, and obviously you're aware this is embarrassing, the person is aware of it, and this is embarrassing. Or a case when when the person is unaware that he's in an embarrassing state, but you're aware of it. For example, you're sitting in a library and the pants are already ripped, and you just observe the person who doesn't know that he is in such conditions. Or just a neutral a scene when just a person is taking a book in the library. So it turned out that in both cases, the left anterior insula and the anterior cingulate cortex, the two regions that were implied with pain and empathy to pain, were activated preferentially in these two cases relative to the neutral state. So this feeling of vicarious embarrassment for the other is also mediated through the same regions. Although, to my knowledge, it is not known if these regions are also involved in direct feeling of embarrassment towards the self, probably because it's a bit hard to design an fMRI experiment to test this. So what about the feeling of disgust? So as I showed you before, in Ekman's faces, one of the universal emotions is disgust. So what brain regions mediate this response? So apparently the same two regions that I described before, the anterior cingulate cortex and the anterior insula. And the experiment was as following. So the subjects were either presented with an odorant, a pleasant or a disgusting odorant, or watched movies of others presented with a pleasure or a disgusting odorant and observed their reactions. And it turned out that the brain regions that were active both when the person was feeling the actual disgusting odorant or observing another disgust were the same brain regions that I mentioned, the anterior insula and the anterior cingulate cortex. So there, in these regions, there was sort of a mirror activation for the feeling of disgust in ourselves and the feeling of disgust in others. And this idea of mirror areas that allow us to appreciate a response in another being and compare it to a response in ourselves stems from other findings of mirror neurons that were found in the monkey brain by the group of Giacomo Rizzolatti. So these neurons basically respond when the animal is doing a movement with its hands, in this case a reaching movement, grabbing of a peanut. And when the experimenter was doing the same movement and the monkey just observed this movement without doing any actual motor movement itself. Hence, they were called the mirror neurons, and I will show you a movie. This is from a talk of Giacomo Rizzolatti that I found on YouTube. He actually will describe himself this response of the mirror neurons. So let's see it for 2 minutes.
What you see here is exhausted neuron.
Can you hear?
Fires when the monkey grasps.
Can you hear him in the back?
Are the speakers on max?
Yeah.
You see how repetible it is. You don't need statistic histograms. It's there. Here is the same neuron, but now it's the vision of this neuron which produces the discharge.
So now the experimenter is doing the movement, and the monkey is observing, and the neuron fires.
It doesn't matter if it's the monkey grasping or the person is grasping. You understand why? Because the goal is the same, it's grasping.
Okay, so I apologize for the sound quality, but basically, I think you got the idea that the same neuron is evolved when we actually do the movement or just seeing another doing the same movement. And the mirror neuron system was identified in two different regions, in the frontal mirror neuron region and the parietal mirror neuron region. These two regions are interconnected, and they receive also a strong input from the STS areas that we discussed before with respect to biological motions. So what are the properties of mirror neurons? So one interesting property is that they really react to the actual action of grasping, not just to a pantomime of this action. So if the experimenter is doing a reaching movement and grasping an object, the neuron will fire. But if you do just a pantomime of this movement without actually touching anything, the neuron will not fire that much. Moreover, it's not really only a visual neuron. So, for example, if there is an object and then you suddenly just put a curtain, so the monkey can't see the object, but it knows it's there, and then you do a movement and you grab the object that is occluded behind the curtain, the neuron will still fire. But in a pantomime case, when there is no object and then you put a curtain and then do a pantomime movement, and the monkeys know that there is no object behind the curtain, the neuron will not respond. So it's really not only the movement, but also the goal of the movement that is important. And another findings that emphasizes this notion is that these neurons also respond to the sound of the action. So in case of peanut breaking, there is a characteristic sound. So normally you have a visual and a sound response, and the neuron fires when you just see the experimenter breaking a peanut, or when it gets the peanut, the monkey gets the peanut and breaks it itself. But if the experimenter will use just a silent version of this movement, so the peanut is already broken and it just breaks it visually without eliciting a sound, still the neuron responds, but it will also respond for the sound alone. So it's kind of a multisensory response that produces the activity, drives the activity of this neuron. So what about mirror neuron systems in humans? So in a way, this empathic studies that I showed you before, especially the study with our reaction to disgust, is an example of mirror neuron systems. But in the case of motor responses, it was shown by fMRI in the following experiment. So the subject was either looking at the display in which in the movie somebody was just raising its index finger, or the subject had to imitate the movement. So it was watching the movie and also raising its index finger itself. And in the case of imitation, the response in the mirror neuron regions was stronger than just to observing the action of the other. So these neurons really, these areas really are involved with imitation. And it was also shown in the case of imitation of facial expression, just of the whole face, the eyes or the mouth, that the activity in these areas was stronger in case of imitation than just to observation of these facial movements. So one interesting way to study the mirror neuron system and to which extent it reflects our motor repertoire is to look at individuals with exceptional motor skills. So in this case, expert dancers. So in this study, there were a group of ballet dancers, a group of capoeira dancers, and a control group. And they were presented either with a video of ballet sequence or a capoeira dance, and the activity in the mirror neuron areas was imaged. So it maybe not surprisingly, the ballet dancer responded more to ballet videos than to capoeira videos, and vice versa for the capoeira dancers. The control subjects responded more or less in the same way for both types of movements. So this area really integrates in a way, or suggested to integrate observed actions with an individual personal motor repertoire. And of course it will be interesting also to study how actors, how mirror neurons respond in actors who are professionals in imitations and facial expressions. So last video for today. So we know this feeling that we see somebody being touched, and we almost feel as if we are being touched ourselves. Usually it's an unpleasant emotion, as in the case of insects. And of course, a lot of horror scenes in movies are based on this ability to have this feeling. So how does it happen? Does the same system of mirror neurons allow us to perceive touch in others as if it was our own body or not? So it turned out that there is a mirror neuron system for touch, but it's located in a bit different brain region. It's located in the supplementary somatosensory cortex, which is the cortical brain region involved in perception of touch. So in this experiment, the subject was put in a magnet and either being touched by the experimenter in the leg, or observed a video of somebody else being touched, either from the subjective view angle, as if it's its own legs being touched by a brush, or by an objective view angle that didn't correspond to the view of its legs. And the control conditions were just the same conditions, but without the subject or the character in the video clip being touched. So it's touch versus not touch, either of self or of a video or of a subject in a video. And it turned out that the brain region that I mentioned, the somatosensory cortex, is more active when somebody is being touched versus non-touched, but is also active when we just see somebody else being touched. And this led the famous neurologist Ramachandran to propose that basically this feeling of being touched when we see another person being touched, that as if it's our own body, the only thing that sort of we still know, as in the case of the movies I show, that it's not our body that the tarantula is going on. So what gives the difference? Of course, we have receptors on our skin. If I touch my arm, there is the receptors in my arm tell my arm being touched. If I see somebody else being touched, I know that it's not my arm because my receptors tell me that's not, there was no activity here. But in the case of amputees, in several case studies, he showed that if this information from sensory receptors in the arm is absent, because there is no arm, so the amputees were actually reporting that if they see somebody else being touched in the corresponding arm to the one that was being amputated, they really felt as if it's their own arm being touched. So probably this system really, in normal conditions, allow us both to feel this sensation of touch in other and the sensation of touch in ourselves, with, of course, differentiating between these two conditions. So I would like to end with a broken mirrors hypothesis in autism, as proposed by Ramachandran and others, that this ability to imitate the others is something very important, probably for evolution and the development of language. There is a gestural hypothesis of language development, and in this motor neuron areas were in a way associated with language areas as well. And in the case of autism, when normal versus autistic kids were asked to imitate different facial expressions, these mirror neurons areas were activated in a weaker way in the autistic individuals while performing this imitation task. And the degree of this impairment in activation of motor neurons areas corresponded with the score, the severity scores of the autistic syndrome. And one of the therapies that been reported, and there are several other studies after 2001 that describe this finding, is that children with autism that were playing with an adult and had to imitate its actions were actually displaying more social behavior than another autistic group that didn't experience this imitation task. So this ability to perceive others, have empathy towards the other is really something fundamental to us, and it's something that is very important in perception of different forms of visual arts. So to sum up, we spoke about motion areas, about biological motion areas in the superior temporal sulcus, about face areas like the fusiform face area here, about areas that are associated with empathy and emotional response, the anterior insula, the anterior cingulate cortex, and the amygdala, and about the mirror neuron system, the frontal and the parietal one. And I would like to finish up with this quote by Mark Matuzek. He was saying that, saying the following: so the next time you start tearing up when you witness someone else in pain, or feel the sensation of grief when you see a tragedy on TV, don't turn away or change the channel. Be grateful that your mirror neurons are doing their job, maintaining the hardwired connection between your heart and the rest of the world. So, thank you.